So dear students, in this video I will make you able to understand a concept of sexual reproduction in Spirogyra with reference to maturation and germination of a zygospore. My friends, you know zygospore formation, this is the very important key aspect in the life of a Spirogyra sexual reproduction. So as you know, sexual reproduction in Spirogyra, it is get accomplished by means of a strategy such as the Scalariform of a conjugation and lateral conjugation. So the zygospore, either which is produced by means of a Scalariform of a conjugation, or it may be by the lateral conjugation. By any means, if it is get produced later on, it is get liberated by the destruction or decay of the wall, that is the cell wall of this zygospore consisting cell. It is get broken, ruptured, and in this way the female cell basically is get broken, and through that the zygospores are get liberated. So these zygospores, they sink to the bottom of the pool or water body over there, which is dehydrated actually, and their thick resistant wall, as you have seen in previous few videos, it consists of a exosporium, mesosporium and endosporium. So this thick wall that enable them to withstand for the desiccation, so that is they are considered as a resting spore over there. And of course this mechanism of a zygospore maturation, it will be carried out later on under the favorable conditions only. So till the conditions are unfavorable, during that it remains as a is that a resting spore over there. So they may be get blown by the wind from the bottom of the dried of pond to the another area, and of course prior to the germination this particular condition should be get fulfilled, that there should be adequate amount of water for the germination of the zygospore. And once this condition is get achieved or attained, what happens prior to the germination of this zygospore, which is actually a one only one stage in the life of a Spirogyra which is actually diploid in nature. So such a diploid zygospore, if you'll see that it possesses the nucleus, and that nucleus, it undergoes a very important event that is called as a meiosis. So my friend, you have to remember that diploid nucleus of a zygospore, it undergoes a meiosis when conditions becomes favorable, and basically this type of a meiotic division in a zygospore of a Spirogyra is generally known as a zygotic meiosis. So please remember this point, zygotic meiosis. And of course this meiosis is as usual comprises two successive divergence. So you know the very first diversion or successful diversion you can called, in which first deductive or deduction diversion maybe takes place, where haploid nucleus it is get divided into the you can say that two sorry, diploid nucleus is get divided into the two haploid one. That is why it is called as a deduction divergent phase, and it is followed by the second meiotic division, which is a, as usual normal phase over there. And of course that consists of toward the end of this one, it will be consist total 4 haploid nuclei, as you can see in the photograph. So both the constitute of a meiosis, the stages, that is deduction 1 and deduction 2 toward the end, is actually consisting the 4 haploid nuclei. Now at the end of this meiosis of a zygospore, whatever be protoplast you can see here in the stage C you can call it as, or this particular phase in the photograph, you will notice it that it consists of 4 haploid nuclei, of course, and consequently what happen, out of these 4 haploid nuclei, 3 of them they get aborted. Okay, so as you can see here in the diagram D, only one it is functional, and rest of them they will get aborted over there. So this is very important part. And of course as usually what happens after the meiosis, generally the cytokinesis maybe takes place in majority of the plant species, but here very distinctly it is observed that there is no as such cytokinesis, and this is the point of a use that a remembrance over there. The survival of haploid nucleus, whatever maybe is there, it is considered as a functional nucleus actually. So whatever maybe is get survived here, it is considered as a functional nucleus. And of course this zygospore, which is consisting a survived haploid nucleus, it will be further get germinate to give rise to a single filament. As you can see in the this particular photograph where the diagram F it is there, that germination, and G is also producing the elongated filament over there. So this is what a very first stage of a use, that is sexual reproduction, in which Spirogyra zygospore, it undergoes a maturation stage, and through that it make itself able to get germinate once conditions becomes much more favorable. So how it exactly get germinated, so that is the point of our discussion, and through which a new filament, single filament, how it is get originated, which is basically a haploid in nature. So from diploid zygospore, how haploid filament is getting produced, this is the part of our discussion, and it is nothing but the next stage followed by the after this maturation of a zygospore, which we can call as a germination of a zygospore. Of course this is taking place under the favorable conditions only. So the zygospore with the surviving functional haploid nucleus that germinates only when there is a presence of a water. And as you can see here, this is the functional nucleus consisting the zygospore, which is of course now this zygospore is now considered as a haploid zygospore. In this particular case, the two outermost layer of the thick zygosporic wall, as you can see here, they are get ruptured, and due to this rupturing the outermost two thick wall and bursting them out, what happens, the content having the haploid nucleus within the protoplast, that is actually earlier it is still it is get enclosed within that particular is that innermost layer. And what happens consequently, this innermost layer, it starts to grow out as a long cylindrical germ tube, in which there is a bright green color appear starts to get appear. As you can see here, this is the actual appearance, reappearance of the filament, which is very small, and it starts to show a green coloration. My friends, if you remember in earlier video, when there is a formation of zygospore takes place, at that time the during stratification, the wall and the colorations, it may be get disappear. More often it is a male protoplast consisting chloroplast. That chloroplast, male chloroplast, it is get actually disappeared. So if you remember that video, you can go back and watch it from the links also over there. So you have to remember that the large spiral chloroplast, which is get disappeared in the formation of a gamete and so their subsequent union, in fact. However, what happens in the zygospore contain it, there is a little bit minute granule, which is generally named as a protoplastid or proplastid. Generally it is called as a proplastid, and this proplastid are considered as a instrumental in the mechanism or formation of a new chloroplast actually. So this is the very interesting fact, why decolored how that decolored zygospore, it gives rise to a colorful filament over there. So answer behind is that is a proplastid. So let's move a little bit ahead now. The green germ tube, it starts to get divide transversely. As you can see here, this green tube, it starts to get divide transversely, and it produces two cells or germ links. You can see that a two cell germ link it is get produced, as it is shown in a diagram G. The lowermost cell of this germ link is colorless, of course, or it is having a little bit green coloration, and it has a rhizoidal character actually. Basically it is having a attachment property or rhizoidal is that a character. So the uppermost cell, it is there which is found in a green coloration, and of course it further get divide, that is green color filament, it starts to get divide repeatedly to form a new filament. So the new filament, this particular filament repetitively undergoes the division, that is upper one, and the young filament, thus it is shown very distinct is that character. More often it is having a base and apex, but more distinction it later on get disappear or it is get lost, and this entire filament, it starts to get float freely in water and becomes a free living. So my friends, I hope that you understood this particular mechanism in which how zygospores get matured, and later on it get germinates to gives rise to a stage of a free floating thallus of a Spirogyra. So here is a very beautiful photograph of a new Spirogyra thallus later on, which metamorphoses and gives rise to branchings, and not branching, but it undergoes a successful mechanism of a divisions. But basically it is nothing but a mature, or you can say that a fully grown Spirogyra filament, which is characterized by what may be property as studied since from the cell structure and its details also. So all the structural details can be observed in this new thallus over there, just like as you can see the nucleus would be there, cytoplasm is there. In fact, cell wall is more distinctly found under microscopic observations. Mucilaginous sheath is there. Of course, chloroplast is the characteristic feature which is spirally present over there, and the factory you can call, or starch, which is nothing but starch synthesizing unit, pyrenites, they are well defined over there in this particular filament. So my friends, I hope that you have really enjoyed this lecture series for this reproduction in a Spirogyra. So thank you very much for observing my all these educational contents and watching these videos. So keep watching for the further details also in near future. I'll keep updating you, so please go through these all videos. So thank you very much.